next up we have Axel Loewe of Karlsruhe Institute of Technology, who's going to be talking to us on digital chimeras on the human atria to enable in-silico analysis scale.
Thank you for the introduction, thank you for the invitation, and thanks to Gernot for setting the stage so perfectly, so I can go quickly over a lot of the introductory things. And it's my great pleasure to show some of our work at this intersection of UQ and digital twins, and also at this intersection of AI and digital twins. I'm not sure if you'll see aspects that cover all three of those. But it's really about electrophysiology in the human atria. And the workhorse model we are using for that is this multi-scale electrophysiology, going from the ion channel via the cell to the tissue to the organ. And by representing all of those biological levels of integration by adequate mathematical equations and coupling them across the scales, we can compute the emergent behavior of it. And to have something useful, it's important to realize that we can change model inputs on all of those scales. So from drug effects, from disease-induced remodeling on the cell level, to of course different shapes of the heart, of the bodies, and also tissue microstructure, to make them individual, to make them representative of a specific subpopulation, if we talk about a virtual cohort. And then run simulations to obtain readouts on all of those scales again. And as we heard in the previous talk, those readouts are not only important to get some feeling for what a change in a parameter in the input would do, or to make some prediction for a specific situation, but also to compare it to real readouts from clinics, from other experimental settings. So this tool, I think, can be used, as we've heard a lot, for personalized medicine through those digital twins. But it's also a very controlled and valuable research environment to understand basic physiology and also pathomechanisms. And it can then be applied to the clinical setting once they are understood. And as also presented in the previous talk, be used for optimizing medical device design, and have this really as a controlled benchmark setting. The point I want to focus on more today is this third aspect and this third use case as a scalable research environment. So once we have this setting and can sample from it, we can generate almost arbitrarily big and quality controlled data sets, for example, to use that for training machine learning approaches then. So this digital twin, or maybe digital snapshot aspect is shown here. As heard multiple times during this conference, it's important to realize that it's not just the anatomy that matters. So this is one important axis that we see here from top to bottom, to find the individual shape of the organ, of the body, whatever. But it also matters that the function is different. So an excitation might travel faster across my heart than across yours, or the other way around. So we really need to pinpoint also this functional parameter vector for individual and personalized modeling. Once we've done that, we have this one point in this 2D space that might be representative of the patient we see up here. What can we do with this digital twin? Well, we can introduce a third dimension, an intervention axis, along which we can move then and systematically evaluate different therapeutic options that in the end will also change anatomy and function, but I think it's helpful as a concept to have that as a third axis, and we move this one point along that third axis and evaluate the effects here. And if we do that, we are in the field of personalized medicine, where we can really use this one-to-one matched digital twin, digital snapshot to evaluate different therapeutic options. In the case of human atrial electrophysiology, for example, the effect of different drugs or different ablation strategies. So we and others have done that, and a common question is which ablation strategy is optimal for a specific patient. Where there is consensus is that the first thing you should try is pulmonary vein isolation, and this works nicely in 50 to 70% of the patients. But for those where it doesn't work, there is a lot of approaches out there. If you talk to to 10 clinicians, you'll probably get 5 different answers. And some prefer to add anatomical lesions, so for example, a roof line on the top of the left atrium. Others say it's the functional substrate that matters, and this can be assessed with late gadolinium enhanced magnetic resonance imaging, for example. So we see this IAR, image intensity threshold. We can do endocardial mapping to identify regions where the electrogram amplitude is low, so low voltage guided ablation. Or we could also look at the conduction velocity from activation maps, and then try to ablate those substrates that appear as good targets. And we could build a personalized model and try to induce arrhythmia AF episodes in that, and then only ablate the organizing centers of those arrhythmias that could be induced in that. So how can that be done? Well, we need personalization on the anatomical level, some imaging, and some functional personalization as well, either also through imaging, if we know where the microstructure is, we might need some activation maps, and then do a virtual stress test. And this can be done in a very aggressive manner, much more comprehensively than could be done in clinics. So we could pace, for example, from all those black points here and try to induce an episode. And if this is successful, it looks something like that. So we have a reentrant activation. And then the question, of course, is which interventions do we apply? So we test different ablation strategies, all those that we've seen in the previous slide. Pretend that we can do perfect ablations, so we render all of those tissue completely non-conductive and perfectly transmural scars, and then see whether this helped or not. And if we do this in a cohort of 29 digital twins, where they were functionally personalized and also anatomically, we see very different success rates. So the height of the bars shows the success rate. Green means complete termination of any arrhythmia, and red means conversion to atrial tachycardia, which is supposed to be a much easier problem to treat clinically, so also considered a success. And what we can see is that in this cohort of 29 individualized patients, PVI was not working well. It's also maybe a particularly challenging one. And we do a very aggressive stress testing. So we don't just try to terminate the current episode, but we also see whether we can reinduce or not. And some of those triggers that we pretend to have in the simulation might not even occur clinically. But we can also see that it makes a difference of whether you ablate the substrate as informed by magnetic resonance imaging or low voltage or conduction velocity. If you do it in the digital twin model, it works quite well, and you might say, okay, problem is solved. But no, we have to be aware where we put the fibrosis in this model. So where did we change the tissue microstructure? And of course, we need to have this informed by the model, and then by the imaging, then put it in the model. And if we look at the difference of whether we put the fibrosis in the model where there was low voltage in the clinical data, or whether there was LGE MRI, we see it doesn't make a difference here, because we just ablate where the functional consequence can be seen, but it shows that those two here are basically swapped. So if we can identify the fibrotic substrate precisely and accurately, and our fibrosis model is good, then this substrate-guided ablation appears to be working very well. But it also shows that, just this uncertainty that we have, whether the substrate is used from the LGE MRI or from the low voltage, and this was done in the same patients, can change the success rate from 90% to 50%, and this is a dramatic change clinically. So that's just one example of such an in silico trial where we did build 29 patient individual models. This is a lot of effort. We get a lot of points here, and then we can subject them all to the same interventions to see how that works. And it's a good way of comparing systematically those different approaches. So we had more than 10 approaches here in those 29 patient models, and this is 290 cases already, so you would need a lot more if you want to do that in a controlled fashion clinically, which is a very high number already. So it's a way, but it doesn't scale very well. So if we want to go from 29 to a few hundred, then either we need very good automated pipelines and access to those data, or maybe something where we would not represent an individual patient, but rather a population of patients in a statistical sense. So this is this concept I first was introduced to in the digital twin workshop that Steve organized about a year ago, where we could move more continuously across both those axes. So we don't want to represent an individual patient, but a likely sample from this population, both on the anatomical and on the functional level. And the good news is that this is possible, for example, with statistical shape models that encode the variability across the population, as shown here for hearts and also body shapes. And then with a principal component analysis-based approach, we can have a mean shape of this population, and then also modes of variation that describe how those points move and how their statistical distribution is. And with that, of course, we can sample and generate arbitrary numbers of likely samples from these populations in the anatomical sense. We can also do this functionally by constraining the parameters a bit, and then, for example, looking at readouts. So this is an example where we change the parameters of the electrophysiology and then look at how the distribution of simulated ECG parameters compares to the same ECG parameters in a large clinical data set. And all of that is publicly available, and we can scale it up to several thousand. I think this was 17,000 simulated compared with a few thousand clinical as well. And we can see how their distributions align, or where they also might not align perfectly. And with that kind of approach, we can really scale up. So this is work led by Albert Dazi from Oxford and Blanca's and Alfonso's group, where he went from 29 to 800 virtual patients, and then looked at variability in all those parameters to reproduce several clinical studies that were ongoing or partly also already completed, and could reproduce the clinical success rates with remarkable accuracy. But not only that, with this number of 800, you can then also identify which of the input features were decisive whether an approach will work or not. And he came up with this kind of manual decision tree showing which approach will likely work best in which subgroup of patients. So it's, for example, about the symmetry of the left and the right atrial sizes, and that will determine whether one ablation strategy or another is likely better or not. So this is an example of what we've heard about in ensemble inference already. And one way how this approach of those digital chimeras and moving in a continuous way can enable such studies. As a last example, I want to show another one where we use this for training machine learning algorithms. So we don't end up with a manual decision tree, but maybe some more sophisticated data science concept to identify the patients. And the tricky part often for machine learning is having adequate data sets of adequate size and with high quality labels that are also not too much biased. And the question we wanted to address here is if we could identify the patients in which this pulmonary vein isolation, so the first line therapy is will likely be sufficient and working well. And the ideal tool for that, or the ideal input data, would be an ECG, because it's so widely available, it's non-invasive, it's cheap. And our clinical partners said, can't you use machine learning for that? And we said, yeah, well, we can try. How much data do you have? Those two centers looked in their databases for how many patients they have all the data complete with constant, etc. And they ended up with a little more than 40. So we said, okay, that's nice, but certainly not sufficient to to train and evaluate a machine learning classifier. So we thought maybe the in silico data could be a solution here, because they scale well, they are quantitative, and provide almost arbitrary resolution. So we use this setting to simulate many episodes of atrial fibrillation, covering this variability, and then also computed the body surface potentials. And then in each of those episodes, we added pulmonary vein isolation and checked whether this terminated or it didn't terminate the arrhythmia. And those were the ground truth labels that were then used to train a classifier, in this case a pretty simple decision tree with somewhat understandable features that was trained purely on those synthetic data, and then after that applied on those 41 or evaluated on those 41 clinical test cases. And it gave surprisingly satisfactory specificity, and especially specificity and consistency outcomes for acute termination. So this was really whether it terminated the arrhythmia acutely or not. Similar approaches can also be used for other questions. So we used it to estimate left atrial fibrotic volume fraction from the ECG with synthetic data. We also used hybrid data sets where we enlarged clinical ones with synthetic ones to identify left atrial enlargement from the ECG, and also to identify atrial flutter locations. So I think that simulations in silico data can help to increase data set size and quality by providing new ways of augmenting such data sets, by working towards a balance of classes, and by having good control about the ground truth labels. But of course, the challenge is that we need to be aware which kind of variability we represent in this model. So if our statistical shape model was trained on a specific subpopulation, maybe more men than women, then we should not expect the results to generalize to those as well. So most of those data sets are openly and freely available. I invite you to check them out. Approach me if you have any questions. Much of this work was done on the electrophysiology side with the Open Carb simulator, and also provides a lot of the modeling resources on the webpage if you're interested in that. As mentioned before, also the next user meeting and training workshop will be in Graz in November this year. And I don't want to miss to thank all the people who did that work, also collaborators, funding, and we'll close with inviting you all to Germany in September this year, where a sequence of conferences will happen, and hope to see many of you there. And now I'm hoping for questions. Thank you.
Nice talk, really nice talk. My question regarding the ablation strategy. Do you think that we are in silico or computational modeling being affected by the clinical set of standard ablations rather than trying our strategy, which is what we can see if it's more suitable for ablation or on in silico? What's your thought on that? Like developing an ablation strategy based on computational modeling rather than the ones that we replicate what's happening in clinic. I know that's the end goal. What we're trying to do is match what they do. But what do you think about that?
So is your question whether we should try to limit the classes to the strategies that are already applied in the clinics rather than suggesting something completely new? Yeah. I think this is a very good exercise to establish credibility of the models. I think this work that Albert has done by basically repeating in silico the clinical studies that were done and showing that they give very similar success rates. I think it's a very strong piece of evidence for the credibility of the models. Yeah. And also, I would say probably the most likely way to have clinical impact, and also for informing clinical trials, because I don't think the in silico ones will replace them, but I think they can refine them and help designing them. And then, of course, there's also this idea of really using the digital twin for a personalized prediction, but it's a much more challenging problem, and we need to get it right. We need to get it right for the very specific patient, whereas those in silico trials on the ensemble, we only need to get it right in a statistical sense. Yeah? So I think both of those approaches have value, but we should take an aware decision when to use which one.
Another question, for the ablation strategy, they say based on LGMRI or low voltage or HDF or whatever it is. We've seen in last HRS says some computational people says fat and fibrosis where they overlap, they are most likely to perpetuate articulations better. Target this region could be better than target either of them. So, do you have any work where you studied like the overlapping area between the different substrate?
So we did not look into fat. So this data set was very rich in the sense that we had LGMRI and also very well done voltage mapping and well curated data. We did not look into fat, and of course, yeah, how you incorporate those fibrotic alterations, be it collagen infiltration or fat deposition into the model, does have an influence as well. There's great work from Caroline's group that you're working in.
Thanks very much for the very nice talk. I think you touch on a very important point, which is the combination with machine learning, and there is no escape, so we need to go there for sure. So can I ask you the same question that I asked to Gernot? Where would you invest your energy if you were an early career researcher setting the direction for your professional life now?
I agree with Gernot that this is a very challenging question, and I don't feel in the position to like giving advice to that. But I would say it's about tools and it's about data. So I would invest, and I do invest in trying to build an ecosystem that's as open as possible. And trying to build things that can be used by others, where others can build on, like this idea, like standing on the shoulders of giants. I think this is the only way we can really move forward as a field also. And sometimes this is harder, sometimes this is easier, but this is what I believe will bring the field and also a specific route forward.
10 points for an Isaac Newton quote there. Very good.
Okay, let's thank Axel again.